Hello students, good morning to all of you. As per your syllabus, we will study the animal secondary growth in the stem of three plants. One is the Bohria, second is the Acaranthus, third is Dracena. So first we will want to study about the animal secondary growth. The angiosperms exhibit a great diversity in the arrangement of various tissues, though most of the families exhibit a well-defined pattern of structure and growth categorized in monocots and dicots, but still many of them exhibit a greater or little amount of deviations from the more usual types. These types are referred to as anomalous and are reported both in dicotyledonous as well as monocotyledonous families. Such anomalies are present both in primary as well as the secondary structures of the stem, and some of the most common anomalies are given here or tell you. One that is anomalous primary structure. In this, the first absence of vessels, scattered vascular bundles in dicot. Third is vascular bundles arranged in ring in monocots. Fourth is presence of cortical or medullary bundles. Fifth is abnormal position of xylem or phloem. Second, the anomalous secondary growth in dicotyledons. In this, first is abnormal behavior of cambium. Second is anomalous position of cambium. Third is accessory cambium formation and its activity. Now, first we will study the anomalous secondary growth in Boerhaavia stem. The primary structure of Boerhaavia stem shows outermost single-layered epidermis, which is followed by the hypodermis. The hypodermis is made up of the few-layered colonchimatous cells. Below the hypodermis, cortex is present, which is made up of the parenchymatous tissue, and the cells of this tissue means parenchymatous possess the chlorophyll, and these cells include small intracellular species. Below the cortex, the endodermis is present. Endodermis is distinct, single-layered, and consists of thin-walled tubular cells. Below the endodermis, pericycle is present. Pericycle consists of thin-walled living parenchymatous cells. In some species, the scanchymatous patches are also present in the pericycle. The, the primary vascular bundles are conjoined, collateral, open, and end arch. They are arranged in three concentric rings. The first ring or the innermost ring consists of two largest and oval vascular bundles found in parenchymatous pith. They lie opposite to each other with protoxylem facing towards each other. The second ring or the middle ring consists of 6 to 14 comparatively small bundles. Next, that is the outermost ring or the third ring. which is consist of 15 to 20 or more small vascular bundles. The secondary growth begins with the formation of cambium strips, formation of cambium strips in the intervascular region in outer ring of vascular bundles. These intervascular cambium strips join with the vascular cambium strips, that is, this is the vascular cambium strips, this is the intravascular cambium strips, and to form a complete ring of cambium. This cambium produces secondary xylem. This cambium produces secondary xylem toward the inner side in the vascular region, just above the primary xylem. And parenchymatous cells in the intervascular region, which becomes lignified and form scamatous conjunctive tissue. Towards outer side, the cambium produces storage parenchyma, this is the storage parenchyma, and secondary phloem, this is the secondary phloem. The secondary phloem is formed towards outer side at places opposite to the secondary xylem. The activity of the cambium ring continues for a definite period and then stops its activity. Then the new cambium strips, develop from the parenchymatous cells produced by first cambium ring, produced by the first cambium ring toward the outer side of secondary xylem. That is the second cambium ring. That is the second cambium ring. These strips develop in the intervascular region. These strips join with the new extravascular cambial strips formed from pericycle lying external to the secondary phloem. External to the secondary phloem. Both kinds of cambium strips join to form a second ring of cambium. The activity of this cambium is same as that of first cambium ring. The activity of this second ring of cambium again stops after producing a definite amount of secondary growth, and third cambium ring develops in the same way. About 4 to 5 about 4 to 5 successive cambium rings are produced, giving rise to 4 or 5 concentric 4 to 5 concentric, that is 4 to 5 concentric growth rings. In case of prostate stem, it has been observed that the growth rings are more strongly developed in the lower side as compared to the upper side, whereas these are symmetrical in erect upright stems. Simultaneously, along with the secondary growth by the activity of cambium ring in outer bundles, the secondary growth also occurs in the innermost and middle ring of medullary bundles. in the innermost and middle ring of vascular bundles. Innermost and middle ring of vascular bundles. This secondary growth is confined within the bundles, means limited only within the bundles. The secondary growth is more pronounced in two large vascular bundles in the center of stem. The vascular cambium gives rise to secondary xylem towards inner side and secondary phloem towards outer side, so that the primary phloem get crushed. The outermost protective covering is formed by the activity of cork, which develops in the hypodermal collenchyma. It produces cork toward the outer side and secondary cortex toward the inner side. The cork layers, cork cambium and secondary cortex form the periderm.